This video contains audio content only. There are no visual elements or images accompanying the audio. If you're expecting to see video footage, please note that this is an intentional audio-only presentation. We recommend listening to this content rather than watching the screen. Thank you for your understanding and enjoy the audio. Hello and welcome, science enthusiasts. Today, we're diving into the fascinating world of microRNAs, a revolutionary field that's changing our understanding of genetics and cellular function. Whether you're a seasoned biologist or just curious about the latest scientific discoveries, this video will take you on a journey through the microscopic realm of these tiny yet powerful molecules. MicroRNAs are reshaping our understanding of gene regulation, development, and disease. In this video, we'll explore recent scientific research that's shedding light on these remarkable molecules. We'll cover everything from their basic functions to their potential applications in medicine, all without getting bogged down in technical jargon. By the end of this video, you'll have a solid grasp of what microRNAs are, why they're important, and how they're revolutionizing fields from developmental biology to personalized medicine. So, let's jump right in and start our exploration of these molecular marvels. 1. The RNA revolution. Understanding microRNAs in the cellular orchestra. To truly appreciate the significance of microRNAs, we first need to understand their place in the broader context of cellular function and the various types of RNA molecules. Let's take a moment to explore the fascinating world of RNA and how microRNAs fit into this complex cellular orchestra. In the grand symphony of life, DNA has long been considered the conductor, directing the production of proteins through its messenger, RNA. But in recent years, we've discovered that the cellular orchestra is far more complex, with various types of RNA playing crucial roles beyond simply relaying messages from DNA to protein-making machinery. At the heart of this complexity are non-coding RNAs, RNA molecules that don't encode proteins but instead perform other vital functions in the cell. These include transfer RNAs, tRNAs, that help translate genetic code into proteins, ribosomal RNAs, rRNAs, that form part of the protein-making factories called ribosomes, and a host of regulatory RNAs that fine-tune gene expression. Enter microRNAs, the rising stars of the RNA world. These tiny molecules, typically only about 22 nucleotides long, have emerged as critical regulators of gene expression. They act like conductors in their own right, fine-tuning the cellular symphony by controlling which genes are turned on or off, and to what extent. What makes microRNAs particularly fascinating is their ability to regulate multiple genes simultaneously. A single microRNA can bind to and control several different messenger RNAs, allowing for coordinated regulation of entire networks of genes. This versatility makes microRNAs powerful players in complex biological processes, from embryonic development to disease progression. Moreover, microRNAs have been found to be involved in nearly every cellular process we studied. They play roles in cell division, differentiation, metabolism, and even cell death. In the immune system, they help fine-tune responses to pathogens. In the brain, they're involved in neuroplasticity and memory formation. The more we look, the more we find these tiny molecules having outsized impacts on cellular function. What's particularly intriguing about microRNAs is their conservation across species. Many microRNAs found in humans have counterparts in other animals, plants, and even some viruses. This high degree of evolutionary conservation suggests that microRNAs perform fundamental biological functions that have been maintained throughout millions of years of evolution. The discovery of microRNAs has also forced us to reconsider our understanding of the genome. Regions once dismissed as junk DNA are now recognized as important sources of regulatory RNAs, including microRNAs. This shift in perspective has opened up new avenues for understanding genetic disorders and developing innovative therapies. In the context of cellular communication, microRNAs have revealed themselves as important messengers. They can be packaged into small vesicles and released from cells, traveling through bodily fluids to influence the function of distant cells. This intercellular communication via microRNAs adds another layer of complexity to our understanding of how organisms coordinate biological processes across tissues and organs. As we delve deeper into the world of microRNAs, we're continually amazed by their versatility and importance. They represent a new frontier in molecular biology, offering fresh insights into how cells regulate their activities and communicate with one another. The study of microRNAs is not just expanding our basic understanding of biology, it's also opening up exciting new possibilities for diagnosing and treating diseases. In the sections that follow, we'll explore the journey of discovery that led to our current understanding of microRNAs, dive into the mechanisms by which they regulate genes, and investigate their roles in health and disease. We'll also look at how researchers are harnessing the power of microRNAs for medical applications and peek into the future of this rapidly evolving field. So, as we move forward, keep in mind that microRNAs are not just another component in the cell. They're key players in the intricate dance of molecular biology, helping to orchestrate the complex symphony of life itself. 2. From discovery to function. Unveiling the world of microRNAs. The story of microRNAs is a testament to the serendipitous nature of scientific discovery and the power of curiosity-driven research. Let's journey through the fascinating history of these tiny molecules and explore how they're made and what they do in our cells. Our tale begins in the early 1990s with a small, unassuming worm. Researchers studying the development of the nematode Caenorhabditis elegans, a favorite model organism in biology, noticed something peculiar. They found that a gene called LIN4, which was known to be important for the worm's development, didn't seem to code for a protein. Instead, it produced a small RNA molecule. At first, this discovery was met with skepticism. The central dogma of molecular biology at the time held that genetic information flowed from DNA to RNA to protein. The idea of a functional RNA that didn't code for a protein was novel and somewhat controversial. But as often happens in science, what seemed like an oddity at first turned out to be the tip of the iceberg. A few years later, researchers discovered another small RNA in C. elegans, called LET7. Unlike LIN4, LET7 was found to be conserved across many animal species, including humans. This was a eureka moment. It suggested that these small RNAs weren't just a quirk of worm biology, but might be part of a fundamental regulatory system in animals. The floodgates opened, and soon researchers were finding these small RNAs, now dubbed microRNAs, in virtually every plant and animal they looked at. The human genome alone was found to contain over 1,000 genes coding for microRNAs, and it's estimated that microRNAs regulate the expression of more than half of all human genes. But how are these microRNAs made? The biogenesis of microRNAs is a multi-step process that begins in the nucleus of the cell. It starts with the transcription of a microRNA gene into a long primary transcript called a pre-mRNA. This pre-mRNA is then processed in the nucleus by an enzyme complex called the microprocessor, which includes the proteins DRASA and DGCR8. The microprocessor clips the pre-mRNA into a shorter molecule called a pre-mRNA, which has a characteristic hairpin structure. This pre-mRNA is then exported from the nucleus to the cytoplasm by a protein called exportin 5. Once in the cytoplasm, the pre-mRNA encounters another enzyme called dicer. Dicer lives up to its name, slicing the pre-mRNA into a short, double-stranded RNA molecule. One strand of this molecule becomes the mature microRNA, while the other strand is typically degraded. The mature microRNA is then loaded into a protein complex called RISC, RNA-induced silencing complex. This microRNA-loaded RISC is the functional unit that goes on to regulate gene expression. But how exactly do microRNAs regulate genes? The key lies in their ability to recognize and bind to specific messenger RNAs, mRNAs. The microRNA acts as a guide, leading the RISC complex to mRNAs that have sequences complementary to the microRNA. Once bound to an mRNA, the microRNA can regulate gene expression in several ways. Most commonly, it represses the translation of the mRNA into protein or triggers the degradation of the mRNA. In some cases, microRNAs can even enhance gene expression, although this is less common. What's particularly interesting about microRNAs is their versatility. A single microRNA can regulate hundreds of different genes, and conversely, a single gene can be regulated by multiple microRNAs. This creates a complex web of regulation that allows for fine-tuning of gene expression in response to various cellular conditions. The discovery of microRNAs has profoundly changed our understanding of gene regulation. We now know that the control of gene expression is far more complex and nuanced than we once thought. MicroRNAs add a layer of regulation that allows cells to rapidly and reversibly adjust their gene expression in response to changing conditions. Moreover, the study of microRNAs has shed light on many biological processes. We've found that microRNAs play crucial roles in development, helping to coordinate the complex series of events that turn a single cell into a fully formed organism. They're involved in maintaining stem cells and in controlling cell differentiation. In the immune system, they help fine-tune immune responses. In the brain, they're involved in processes like neuroplasticity and memory formation. The more we study microRNAs, the more we appreciate their importance in biology. They're not just passive players in cellular processes, they're active regulators, helping to orchestrate the complex symphony of molecular events that make life possible. As we move forward in our exploration of microRNAs, we'll delve deeper into how they regulate genes, their roles in health and disease, and how we might be able to harness their power for medical applications. The world of microRNAs is a frontier of modern biology, full of exciting discoveries and potential applications. So let's continue our journey into this fascinating molecular landscape. 3. The molecular maestros. How microRNAs conduct gene regulation. Now that we've explored the discovery and biogenesis of microRNAs, let's dive deeper into how these tiny molecules orchestrate gene regulation. It's a fascinating process that showcases the elegance and complexity of cellular biology. At its core, the function of microRNAs is to fine-tune gene expression. They act like dimmer switches, able to dial the expression of their target genes up or down as needed. This ability to make subtle adjustments to gene expression is crucial for maintaining cellular homeostasis and responding to changing conditions. The key to understanding microRNA function lies in their ability to recognize and bind to specific messenger RNAs, mRNAs. Each microRNA has a specific sequence that allows it to pair with complementary sequences in the mRNAs of its target genes. This sequence-specific targeting is what gives microRNAs their power and specificity. The region of the microRNA that's most important for target recognition is called the seed sequence. This is typically a stretch of 6 to 8 nucleotides near the 5' end of the microRNA. The seed sequence binds to complementary sites in the target mRNA, usually located in the 3' untranslated region, 3' UTR. Once a microRNA finds its target mRNA, it can regulate gene expression in several ways. The most common mechanism is translational repression coupled with mRNA destabilization. In this scenario, the microRNA RISC complex binds to the target mRNA and recruits proteins that inhibit translation and promote mRNA decay. One of the fascinating aspects of microRNA-mediated regulation is its scalability. A single microRNA can have hundreds of target mRNAs, allowing for coordinated regulation of entire networks of genes. This makes microRNAs powerful tools for controlling complex biological processes. Moreover, the regulation by microRNAs is often combinatorial. A single mRNA can have binding sites for multiple different microRNAs, and the combined effect of these microRNAs determines the final level of gene expression. This allows for extremely fine-tuned control of gene expression in response to various cellular signals. But the story doesn't end there. Recent research has revealed that microRNA-mediated regulation is even more complex than we initially thought. For instance, we now know that some microRNAs can actually enhance gene expression under certain conditions, contrary to their typical repressive function. Another layer of complexity comes from the fact that microRNA activity can be modulated by various factors. The availability of microRNA binding sites can be affected by RNA binding proteins or by alternative splicing of mRNAs. Some mRNAs even have sites that can sequester microRNAs, acting as sponges to reduce the activity of specific microRNAs. The cellular localization of microRNAs also plays a role in their function. While most microRNA-mediated regulation occurs in the cytoplasm, some microRNAs have been found to function in the nucleus, where they can regulate gene expression at the transcriptional level. One of the most exciting recent discoveries in the field of microRNA research is the role of these molecules in cell-to-cell communication. MicroRNAs can be packaged into small vesicles called exosomes and released from cells. These exosomes can then be taken up by other cells, allowing the microRNAs to regulate gene expression in their new host cells. This mechanism of intercellular communication adds yet another layer to the complex regulatory networks controlled by microRNAs. The importance of microRNAs in gene regulation becomes clear when we look at what happens when microRNA function is disrupted. Knockout studies in various organisms have shown that loss of specific microRNAs can lead to severe developmental defects or increased susceptibility to disease. This underscores the crucial role that microRNAs play in maintaining proper cellular function. In recent years, researchers have also begun to appreciate the role of microRNAs in cellular stress responses. Many microRNAs are upregulated in response to various forms of cellular stress, where they help to coordinate the cell's adaptive response. This ability to rapidly modulate gene expression in response to stress makes microRNAs important players in cellular resilience. The study of microRNA-mediated gene regulation has also led to new insights into the evolution of gene regulatory networks. Many microRNAs and their target sites are conserved across species, suggesting that these regulatory relationships have been maintained over millions of years of evolution. At the same time, the emergence of new microRNAs may have played a role in the evolution of complex traits and the diversification of species. As our understanding of microRNA-mediated gene regulation grows, so too does our appreciation for the complexity of cellular biology. These tiny RNA molecules, once thought to be quirks of worm genetics, are now recognized as master regulators of gene expression, playing crucial roles in virtually every aspect of cellular function. Looking ahead, the field of microRNA research continues to evolve rapidly. New technologies are allowing us to study microRNA function with unprecedented precision, and we're constantly discovering new roles for these versatile molecules. From development to disease, from stress responses to intercellular communication, microRNAs are proving to be central players in the intricate dance of cellular biology. In the next section, we'll explore how this intricate system of microRNA-mediated gene regulation impacts health and disease, paving the way for new understanding and potential therapeutic interventions. The world of microRNAs is a frontier of modern biology, full of exciting discoveries and potential applications. So let's continue our journey into this fascinating molecular landscape. 4. MicroRNAs in health and disease, tiny players with big impacts. As we've explored the intricate world of microRNAs and their role in gene regulation, you might be wondering, what does all this mean for human health and disease? The answer, as it turns out, is quite a lot. MicroRNAs have emerged as key players in a wide range of physiological processes and pathological conditions, making them not only important subjects of basic research but also promising targets for medical interventions. Let's start by considering the role of microRNAs in normal physiological processes. From the moment of conception, microRNAs are hard at work, helping to guide embryonic development. They play crucial roles in cell differentiation, helping to determine the fate of stem cells and ensure that different cell types develop in the right place at the right time. In the adult organism, microRNAs continue to play important roles in maintaining tissue homeostasis. They're involved in regulating cell division, metabolism, and programmed cell death. In the immune system, microRNAs help fine-tune immune responses, ensuring that the body can fight off pathogens effectively while avoiding excessive inflammation. MicroRNAs are also important players in the nervous system. They're involved in neuroplasticity, the brain's ability to form and reorganize synaptic connections. This makes them crucial for processes like learning and memory formation. Some microRNAs even show circadian rhythms of expression, suggesting they may play a role in regulating our biological clocks. Given their importance in normal physiology, it's not surprising that dysregulation of microRNAs can contribute to disease. In fact, altered microRNA expression has been observed in virtually every disease that has been studied, from cancer to cardiovascular disease to neurological disorders. Let's take a closer look at the role of microRNAs in cancer, as this has been one of the most intensively studied areas of microRNA research. Many microRNAs have been found to act as tumor suppressors, helping to prevent uncontrolled cell growth. When these microRNAs are down-regulated or lost, it can contribute to cancer development. For example, the LET7 family of microRNAs, which we mentioned earlier as one of the first microRNAs discovered, has been found to act as a tumor suppressor in many types of cancer. LET7 microRNAs target several oncogenes, including ROS, which is frequently mutated in cancer. Loss of LET7 expression is associated with poor prognosis in several types of cancer. On the flip side, some microRNAs can act as oncogenes when they're overexpressed. These oncomers promote cancer development by targeting tumor suppressor genes. A well-known example is MIR21, which is overexpressed in many types of cancer and promotes cell proliferation, invasion, and metastasis. The role of microRNAs in cancer isn't limited to tumor cells themselves. MicroRNAs also play important roles in the tumor microenvironment, influencing processes like angiogenesis, the formation of new blood vessels, and immune evasion. This makes microRNAs potential targets for cancer therapies that aim to modify the tumor microenvironment. But cancer is far from the only disease where microRNAs play a role. In cardiovascular disease, microRNAs have been found to regulate processes like cardiac remodeling, fibrosis, and angiogenesis. Some microRNAs, like MIR208A, have been proposed as potential biomarkers for heart attack. In neurodegenerative diseases like Alzheimer's and Parkinson's, dysregulation of specific microRNAs has been observed. These microRNAs may contribute to disease progression by affecting processes like protein aggregation and neuroinflammation. For example, MIR34A has been found to be upregulated in Alzheimer's disease and may contribute to neural cell death. MicroRNAs also play important roles in metabolic diseases. In diabetes, for instance, microRNAs are involved in regulating insulin secretion and sensitivity. MIR375, for example, is highly expressed in pancreatic islet cells and helps regulate insulin secretion. Even in infectious diseases, microRNAs play a role. Host microRNAs can influence susceptibility to infection and the intensity of the immune response. Moreover, some viruses have evolved to produce their own microRNAs or to hijack host microRNAs to promote their replication. One particularly interesting area of microRNA research in disease is the study of circulating microRNAs. These are microRNAs that can be detected in bodily fluids like blood, urine, or saliva. Circulating microRNAs have generated a lot of excitement as potential biomarkers for various diseases. The idea is that the profile of microRNAs in a person's blood, for example, might provide information about their health status or the presence of disease. This could potentially lead to new, non-invasive diagnostic tests. For instance, specific patterns of circulating microRNAs have been associated with different types of cancer, raising the possibility of liquid biopsies that could detect cancer through a simple blood test. However, it's important to note that while the potential of microRNAs in disease diagnosis and treatment is exciting, many challenges remain. The complex regulatory networks involving microRNAs mean that manipulating a single microRNA can have wide-ranging and sometimes unpredictable effects. Moreover, delivering microRNA-based therapies to specific tissues in the body remains a significant technical challenge. Despite these challenges, the study of microRNAs in health and disease continues to yield fascinating insights. We're learning that these tiny RNA molecules play outsized roles in countless biological processes, from embryonic development to aging. They're involved in maintaining health and in driving disease, making them both important subjects of basic research and promising targets for medical intervention. As we continue to unravel the complex world of microRNAs, we're gaining a deeper understanding of cellular biology and the molecular basis of disease. This knowledge is not just academically interesting, it has the potential to transform medicine, leading to new diagnostic tools and innovative therapies. In our next section, we'll explore some of the ways researchers are working to harness the power of microRNAs for medical applications. From diagnostics to therapeutics, microRNAs are opening up exciting new possibilities in the world of medicine. So let's continue our journey into the frontier of microRNA research and its potential to revolutionize healthcare. 5. The promise of microRNA medicine, diagnostics and therapeutics. As our understanding of microRNAs and their roles in health and disease has grown, so too has interest in harnessing these molecules for medical applications. From developing new diagnostic tools to designing innovative therapies, microRNAs are at the forefront of a new era in personalized medicine. Let's explore some of the exciting developments in this rapidly evolving field. First, let's consider the potential of microRNAs in diagnostics. As we mentioned earlier, circulating microRNAs, those found in bodily fluids like blood, urine, or saliva, have generated a lot of excitement as potential biomarkers for various diseases. The idea is compelling. A simple blood test that could detect the presence of disease or predict its progression based on the profile of microRNAs present. One area where this approach shows particular promise is in cancer diagnostics. Different types of cancer have been associated with specific patterns of circulating microRNAs. For example, a panel of serum microRNAs has been identified that can distinguish patients with pancreatic cancer from healthy individuals with high accuracy. Similar approaches are being explored for other types of cancer, as well as for cardiovascular diseases, neurodegenerative disorders, and more. The potential advantages of microRNA-based diagnostics are numerous. They could allow for earlier detection of disease, when treatment is often more effective. They're non-invasive, requiring only a blood sample rather than a tissue biopsy. And because microRNAs are relatively stable in blood, these tests could potentially be more robust than those based on other types of molecules. However, it's important to note that while the potential is exciting, microRNA-based diagnostics are still largely in the research phase. Standardizing methods for measuring circulating microRNAs and establishing reliable reference ranges are ongoing challenges. Nevertheless, the field is advancing rapidly, and it's likely that we'll see microRNA-based diagnostic tests becoming available in the clinic in the coming years. But diagnostics is just one side of the coin. Perhaps even more exciting is the potential of microRNAs as therapeutic agents. The basic idea is simple. If dysregulation of specific microRNAs contributes to disease, then correcting that dysregulation could potentially treat the disease. There are two main approaches to microRNA-based therapeutics. The first is to supplement microRNAs that are down-regulated in disease. This approach, known as microRNA replacement therapy, involves delivering synthetic microRNAs or microRNA mimics to restore the function of tumor suppressor microRNAs, for example. The second approach is to inhibit microRNAs that are overexpressed in disease. This can be done using anti-microRNA oligonucleotides, AMOS, or microRNA sponges, which bind to and sequester the target microRNA, preventing it from regulating its target genes. Both approaches have shown promise in preclinical studies. For example, delivery of LET7 mimics has been shown to inhibit tumor growth in animal models of lung cancer. On the flip side, inhibition of MIR122, a microRNA involved in hepatitis C virus replication, has shown promise as a treatment for hepatitis C in clinical trials. One of the exciting aspects of microRNA-based therapeutics is their potential to address complex diseases by targeting multiple genes simultaneously. Because a single microRNA can regulate hundreds of genes, modulating the level of one microRNA could potentially affect entire gene networks. This multi-target approach could be particularly valuable for diseases like cancer, where multiple pathways are typically dysregulated. However, as with any new therapeutic approach, there are challenges to overcome. One major hurdle is delivery. Getting the microRNA or anti-microRNA to the right cells in the body. RNA molecules are quickly degraded in the bloodstream and don't easily cross cell membranes. Researchers are exploring various delivery systems, including nanoparticles and viral vectors, to overcome these obstacles. Another challenge is off-target effects. Because microRNAs regulate many genes, altering the level of a single microRNA could potentially have wide-ranging effects throughout the body. Careful design of microRNA-based therapeutics and targeted delivery systems will be crucial to minimize these off-target effects. Despite these challenges, the field of microRNA therapeutics is advancing rapidly. Several microRNA-based drugs are currently in clinical trials for various diseases, including cancer, hepatitis C, and cardiovascular disease. While it's still early days, the results so far are encouraging. Beyond direct therapeutic applications, microRNAs are also proving valuable in drug development. Understanding how existing drugs affect microRNA expression can provide insights into their mechanisms of action and potential side effects. Moreover, microRNA expression profiles are being explored as a way to predict patient response to specific treatments, paving the way for more personalized medicine approaches. As we look to the future, the potential applications of microRNAs in medicine seem almost limitless. From early disease detection to personalized treatments, microRNAs could revolutionize how we approach healthcare. They offer the promise of more precise diagnostics, more effective treatments, and a deeper understanding of the molecular basis of disease. Of course, realizing this potential will require continued research and development. We need to deepen our understanding of microRNA biology, develop more effective delivery methods for microRNA-based therapeutics, and conduct rigorous clinical trials to ensure safety and efficacy. But the pace of progress in this field is truly exciting. Each year brings new discoveries about the roles of microRNAs in health and disease, and new advances in our ability to harness these tiny molecules for medical applications. As we continue to unlock the secrets of microRNAs, we're opening up new frontiers in personalized medicine and bringing closer the dream of more effective, targeted treatments for a wide range of diseases. In our next section, we'll explore some of the cutting-edge technologies that are driving advances in microRNA research, particularly focusing on methods for detecting and analyzing extracellular microRNAs. These technological innovations are not only advancing our scientific understanding but also bringing us closer to practical applications of microRNA research in medicine and beyond. So let's continue our journey into the fascinating world of microRNA research and its potential to transform healthcare. 6. Beyond the cell. Extracellular microRNAs and cutting-edge detection methods. As our understanding of microRNAs has grown, one of the most exciting developments has been the discovery of extracellular microRNAs. These are microRNAs that are found outside of cells, circulating in bodily fluids like blood, urine, and saliva. This discovery has opened up new avenues for research and potential clinical applications, particularly in the realm of diagnostics. Let's explore the world of extracellular microRNAs and the cutting-edge methods being developed to detect and analyze them. The existence of stable microRNAs outside of cells was initially surprising to researchers. RNA molecules are typically quite fragile and are quickly degraded when exposed to the enzymes present in bodily fluids. However, extracellular microRNAs have been found to be remarkably stable, able to withstand conditions that would destroy most other RNA molecules. This stability is due to the way extracellular microRNAs are packaged and transported. They can be found in several different forms in the extracellular environment. Some are enclosed within small vesicles called exosomes, which protect them from degradation. Others are associated with protein complexes, particularly with proteins from the argonaute family, which are key players in microRNA function inside cells. The discovery of extracellular microRNAs has led to some intriguing questions. Why are these microRNAs being released from cells? Do they have a functional role in cell-to-cell communication? And perhaps most excitingly from a medical perspective, can the profile of extracellular microRNAs provide information about the health status of an individual? Research has shown that cells do indeed appear to selectively release certain microRNAs, and that these extracellular microRNAs can be taken up by other cells and influence their function. This suggests a new mode of cell-to-cell communication, with microRNAs acting as signaling molecules that can transmit information between distant cells in the body. In terms of medical applications, the potential of extracellular microRNAs as biomarkers has generated a lot of excitement. The idea is that the profile of microRNAs in a person's blood, for example, might provide a snapshot of their health status. Changes in this profile might indicate the presence of disease, potentially allowing for early detection of conditions like cancer or heart disease. However, detecting and accurately measuring extracellular microRNAs presents some unique challenges. These molecules are present in very low concentrations in bodily fluids, and accurately quantifying them requires highly sensitive and specific methods. This has driven the development of new, cutting-edge detection technologies. One of the most widely used methods for detecting microRNAs is quantitative PCR, qPCR. This technique amplifies specific microRNA sequences, allowing them to be detected and quantified. While qPCR is sensitive and specific, it requires prior knowledge of the microRNA sequences you're looking for, and it can be time-consuming when analyzing many different microRNAs. To address these limitations, researchers have developed microarray-based methods that can detect hundreds or even thousands of different microRNAs simultaneously. These microarrays consist of tiny spots of DNA probes that can bind to specific microRNAs. By measuring how much of each probe binds to microRNAs in a sample, researchers can get a comprehensive picture of which microRNAs are present and in what quantities. Perhaps the most powerful tool for microRNA analysis is next-generation sequencing, NGS. This technology allows researchers to sequence all the microRNAs in a sample, providing a complete profile of microRNA expression. NGS has the advantage of being able to detect novel microRNAs and to distinguish between closely related microRNA sequences. However, it requires sophisticated bioinformatics tools to analyze the large amounts of data generated. In recent years, there's been growing interest in developing point-of-care tests for microRNA detection. These are tests that could potentially be performed quickly and easily, perhaps even in a doctor's office or at home. One promising approach is the use of electrochemical biosensors, which can detect microRNAs by measuring changes in electrical current when the microRNAs bind to specific probes. Another innovative approach is the use of nanomaterials for microRNA detection. For example, some researchers have developed gold nanoparticles that change color when they bind to specific microRNAs, potentially allowing for visual detection of microRNAs without the need for sophisticated equipment. As detection methods have improved, so too have methods for isolating and purifying extracellular microRNAs from bodily fluids. This is a crucial step in any analysis, as the quality and purity of the isolated microRNAs can significantly affect the results of downstream analyses. Researchers have developed various protocols for isolating microRNAs from different types of samples, including methods specifically designed to isolate exosome-associated microRNAs. One particularly exciting development in this area is the use of microfluidic devices for isolating and analyzing extracellular microRNAs. These devices use tiny channels and chambers to manipulate small volumes of fluid, allowing for efficient isolation and analysis of microRNAs from very small samples. Some microfluidic devices integrate multiple steps of the analysis process, from sample preparation to detection, potentially allowing for rapid, automated microRNA analysis. As these technologies continue to advance, they're not only improving our ability to study extracellular microRNAs but also bringing us closer to practical applications in medicine. For example, researchers are working on developing blood tests based on extracellular microRNA profiles that could detect cancer at early stages or predict the risk of heart disease. However, it's important to note that there are still challenges to overcome before extracellular microRNA analysis can be widely used in clinical settings. One major issue is standardization. Different labs often use different methods for isolating and measuring microRNAs, making it difficult to compare results across studies. Efforts are underway to develop standardized protocols and reference materials to address this issue. Another challenge is understanding the biological significance of changes in extracellular microRNA profiles. While many studies have found associations between certain microRNA patterns and various diseases, we still have much to learn about what these patterns mean and how they relate to underlying biological processes. Despite these challenges, the field of extracellular microRNA research is advancing rapidly. Each year brings new discoveries about the roles of these molecules in health and disease, and new technologies for detecting and analyzing them. As we continue to refine our methods and deepen our understanding, extracellular microRNAs hold great promise as both tools for basic research and as the basis for new diagnostic and therapeutic approaches. In our next section, we'll take a step back and consider the evolutionary significance of microRNAs. How have these tiny regulators shaped the development of complex life over millions of years? And what can studying the evolution of microRNAs tell us about their functions today? Let's continue our exploration of the fascinating world of microRNAs. 7. Nature's conserved language. The evolutionary significance of microRNAs. As we've journeyed through the world of microRNAs, we've seen how these tiny molecules play crucial roles in gene regulation, development, and disease. But to truly appreciate the significance of microRNAs, we need to consider them from an evolutionary perspective. How did these regulatory molecules arise? How have they changed over time? And what can their evolutionary history tell us about their functions today? Let's explore the fascinating story of microRNA evolution. The evolutionary history of microRNAs is a testament to their fundamental importance in biology. Many microRNAs are highly conserved across species, meaning that the same or very similar microRNAs can be found in organisms as diverse as humans, flies, and even plants. This high degree of conservation suggests that these microRNAs play essential roles that have been maintained by natural selection over hundreds of millions of years of evolution. Let's start at the beginning. The first microRNAs likely evolved over 1 billion years ago, before the split between plants and animals. These ancient microRNAs were probably involved in basic cellular processes that are common to all complex life forms. As plants and animals diverged and evolved separately, they each developed their own unique sets of microRNAs in addition to the conserved, ancient ones. In animals, there was a significant expansion in the number of microRNAs coinciding with the evolution of more complex body plans. For instance, there was a substantial increase in microRNA diversity during the evolution of vertebrates, and again during the evolution of mammals. This suggests that the expansion of microRNA repertoires may have played a crucial role in the evolution of complex traits and developmental processes. One of the fascinating aspects of microRNA evolution is how new microRNAs arise. There are several mechanisms by which this can happen. Sometimes, a gene encoding a microRNA can be duplicated, leading to two copies that can then diverge and take on new functions. In other cases, random sequences in the genome can evolve into functional microRNAs if they happen to form the characteristic hairpin structure and are transcribed. Interestingly, many microRNAs are found in clusters in the genome, suggesting that they may have arisen through duplication events and then diversified over time. These clustered microRNAs often work together to regulate related processes, providing an elegant example of how evolution can produce coordinated regulatory systems. The evolutionary history of microRNAs is written not just in their sequences, but also in the sequences of their target sites in mRNAs. Many microRNA binding sites are also highly conserved, indicating that the regulatory relationships between microRNAs and their targets have been maintained over long evolutionary timescales. This conservation of target sites provides additional evidence for the functional importance of microRNA-mediated regulation. However, it's important to note that while many microRNAs are highly conserved, others are species-specific or limited to closely related groups of organisms. These more recently evolved microRNAs may be involved in regulating traits that are specific to particular lineages. For example, some microRNAs found only in primates may play roles in aspects of brain development that are unique to our lineage. the study of microRNA evolution has provided some intriguing insights into broader evolutionary processes. For instance, it appears that the evolution of new microRNAs may have contributed to the evolution of new cell types and developmental processes. This suggests that changes in gene regulation, rather than just changes in protein-coding genes, have played a crucial role in the evolution of complex life. Moreover, the evolutionary history of microRNAs may help explain some of the complexities of gene regulation that we observe today. For example, the fact that many genes are regulated by multiple microRNAs, and that many microRNAs regulate multiple genes, may be a result of the gradual accumulation of regulatory relationships over evolutionary time. Understanding the evolution of microRNAs is not just of academic interest, it has practical implications as well. For instance, highly conserved microRNAs often regulate fundamental biological processes and may therefore be good targets for therapeutic interventions. On the other hand, species-specific microRNAs might be involved in traits that are unique to particular organisms, which could be important to consider when using animal models to study human diseases. The evolutionary perspective also provides valuable context for interpreting the results of microRNA studies. For example, if a microRNA is highly conserved across species, we might expect it to have a crucial function that's relevant to many different organisms. Conversely, if a microRNA is only found in a particular group of organisms, it might be involved in traits specific to that group. As we look to the future, the study of microRNA evolution continues to yield fascinating insights. New sequencing technologies are allowing researchers to discover microRNAs in a wider range of organisms, providing a more comprehensive picture of how these regulatory molecules have evolved. At the same time, experimental studies are shedding light on how changes in microRNA sequences or expression patterns can contribute to evolutionary changes in gene regulation and organismal traits. One particularly intriguing area of research is the role of microRNAs in rapid evolutionary adaptation. There's evidence that changes in microRNA expression or target sites can allow organisms to quickly adapt to new environments or stresses. This suggests that microRNAs might play a crucial role in how species respond to environmental changes, including those caused by human activities. Another exciting frontier is the study of how microRNAs might have contributed to major evolutionary transitions, such as the origin of multicellularity or the evolution of complex developmental processes. By comparing the microRNA repertoires of different organisms and studying how they regulate gene expression, researchers are gaining new insights into how complex life evolved. The evolutionary story of microRNAs is a testament to the power of small changes to have big effects. These tiny RNA molecules, through their ability to fine-tune gene expression, have played a crucial role in shaping the diversity of life on Earth. As we continue to unravel their evolutionary history, we're not just learning about the past, we're gaining valuable insights that can inform our understanding of biology today and guide future research and medical applications. In our final section, we'll look towards the future of microRNA research. What are the biggest challenges and opportunities in this field? How might advances in microRNA research shape biology and medicine in the coming years? Let's explore the frontiers of this exciting field. 8. Frontiers of microRNA research, challenges and future horizons. As we've journeyed through the world of microRNAs, we've seen how these tiny molecules play outsized roles in biology and medicine. But like any frontier of science, the field of microRNA research is full of both challenges and opportunities. In this final section, let's explore some of the key questions and directions that are shaping the future of microRNA research. One of the most pressing challenges in the field is to fully elucidate the complex networks of microRNA-mediated regulation in cells. While we've made great strides in identifying microRNAs and their targets, understanding how all these interactions come together to control cellular processes is a formidable task. It's like trying to understand a symphony by studying each instrument in isolation. We need to figure out how all the parts work together. Advances in systems biology approaches are helping to address this challenge. Researchers are developing sophisticated computational models that can integrate data on microRNA expression, target predictions, and cellular outcomes to build more comprehensive pictures of microRNA-mediated regulation. These models, combined with experimental validation, are starting to reveal the emergent properties of microRNA regulatory networks. Another frontier is understanding the dynamics of microRNA regulation. Most studies to date have provided static snapshots of microRNA expression and activity. But cells are constantly changing, responding to internal and external signals. How do microRNA regulatory networks change over time, for example during development or in response to environmental stresses? New technologies that allow for real-time monitoring of microRNA activity in living cells are starting to provide insights into these dynamics. The role of microRNAs in intercellular communication is another exciting area of research. As we discussed earlier, microRNAs can be packaged into exosomes and transported between cells. But many questions remain. How specific is this process? Can cells selectively package and send particular microRNAs to other cells? And how do recipient cells interpret these microRNA signals? Understanding these processes could have important implications for our understanding of how tissues and organs coordinate their activities. In the medical realm, one of the big challenges is translating our growing knowledge of microRNAs into effective therapies. While there's been progress in developing microRNA-based drugs, delivering these molecules to specific tissues in the body remains a significant hurdle. Researchers are exploring various approaches, from nanoparticle-based delivery systems to engineered viruses, but much work remains to be done to make these therapies safe and effective. Another challenge in the therapeutic realm is ensuring the specificity of microRNA-based interventions. Because each microRNA can regulate hundreds of genes, altering the levels of a single microRNA could potentially have wide-ranging effects throughout the body. Developing ways to target microRNA therapies to specific tissues or cell types will be crucial for minimizing off-target effects. On the diagnostic front, while the potential of microRNAs as biomarkers is exciting, there are still hurdles to overcome before microRNA-based tests can be widely used in the clinic. Standardizing methods for measuring microRNAs in biological samples and establishing reliable reference ranges are ongoing challenges. Moreover, we need to better understand how changes in circulating microRNA profiles relate to specific disease states. Looking further ahead, there are some intriguing possibilities on the horizon. For instance, could we use our understanding of microRNA regulation to design synthetic gene circuits for use in biotechnology or medicine? Could we engineer new microRNAs to regulate genes in ways that don't occur naturally? These kinds of approaches could open up new possibilities for controlling cellular behavior. Another frontier is understanding the role of microRNAs in complex biological phenomena like aging. There's growing evidence that changes in microRNA expression contribute to the aging process, but many questions remain. Could manipulating microRNA levels help to slow aging or alleviate age-related diseases? This is an area of intense research interest. The intersection of microRNA research with other cutting-edge fields is also opening up new possibilities. For example, CRISPR gene editing technology is providing new tools for studying microRNA function and for potentially correcting disease-causing mutations in microRNA genes or their target sites. Meanwhile, advances in artificial intelligence and machine learning are enhancing our ability to predict microRNA targets and to interpret complex data on microRNA expression and function. As we look to the future, it's clear that the field of microRNA research will continue to evolve rapidly. New technologies will undoubtedly emerge, providing new ways to study these fascinating molecules. At the same time, our growing understanding of microRNA biology will likely lead to new questions and new areas of investigation. One thing seems certain: microRNAs will continue to surprise us. Time and again, research into these tiny RNA molecules has revealed unexpected complexities in cellular biology and opened up new possibilities for understanding and treating disease. As we continue to unlock the secrets of microRNAs, we're not just advancing our scientific knowledge. We're paving the way for new approaches to some of the most pressing challenges in biology and medicine. In conclusion, the story of microRNAs is a testament to the power of basic scientific research. What began as a curious observation in worms has blossomed into a field that touches on virtually every area of biology and medicine. From embryonic development to cancer, from evolution to cutting-edge therapeutics, microRNAs are proving to be key players in the intricate dance of life. As we've seen throughout this exploration, the world of microRNAs is complex and fascinating, full of both challenges and opportunities. Whether you're a scientist working in this field, a student learning about it for the first time, or simply someone curious about the frontiers of biology, there's no doubt that microRNAs will continue to be an exciting area to watch in the years to come. Thank you for joining me on this journey through the world of microRNAs. I hope this exploration has given you a deeper appreciation for these tiny but mighty molecules and the researchers working to understand them. As we continue to unravel the mysteries of microRNAs, who knows what amazing discoveries await us? The frontier of microRNA research is wide open, and the future is full of possibilities. As we wrap up our exploration of microRNAs, let's take a moment to reflect on some of the key points we've covered and consider their broader implications. First and foremost, we've seen that microRNAs, despite their small size, play crucial roles in virtually every aspect of cellular function. From embryonic development to aging, from normal physiology to disease, these tiny regulators are involved in a vast array of biological processes. This underscores the importance of gene regulation in biology. It's not just the genes themselves that matter, but how they're controlled and expressed. We've also seen how the study of microRNAs has transformed our understanding of gene regulation. The discovery of these molecules revealed a whole new layer of control over gene expression, one that allows for fine-tuning and coordination of complex genetic programs. This has profound implications for how we think about the relationship between genotype and phenotype, and how we approach the study of biological systems. The evolutionary conservation of many microRNAs highlights their fundamental importance in biology. The fact that some microRNAs have been conserved across hundreds of millions of years of evolution suggests that they play essential roles that have stood the test of time. At the same time, the evolution of new microRNAs has likely contributed to the development of new traits and capabilities in various lineages. From a medical perspective, the involvement of microRNAs in numerous diseases opens up new avenues for diagnosis and treatment. The potential of microRNAs as biomarkers could lead to new, non-invasive diagnostic tests for a variety of conditions. Meanwhile, therapies based on modulating microRNA activity hold promise for treating diseases at the molecular level. The study of microRNAs also exemplifies the power of interdisciplinary research. Advances in this field have required collaborations between molecular biologists, geneticists, biochemists, bioinformaticians, and many others. As we tackle increasingly complex biological questions, this kind of interdisciplinary approach will become ever more important. Looking ahead, the field of microRNA research is poised to continue making important contributions to biology and medicine. As new technologies emerge and our understanding deepens, we can expect to see new insights into fundamental biological processes, new diagnostic tools, and new therapeutic approaches based on microRNA biology. However, it's important to remember that there's still much we don't know about microRNAs. Many challenges remain, from fully understanding the complex regulatory networks involving microRNAs to developing effective microRNA-based therapies. These challenges represent opportunities for future research and innovation. For those of you watching who are curious about science or considering a career in biology, the field of microRNA research offers a vivid example of how a seemingly small discovery can open up whole new areas of study. It's a reminder that in science, you never know where a line of inquiry might lead. To our viewers who might be dealing with health issues or have loved ones facing medical challenges, the advances in microRNA research offer hope. While many applications are still in the research phase, the growing understanding of microRNAs is paving the way for new approaches to diagnosing and treating a wide range of diseases. For educators and students, the story of microRNAs provides a compelling case study in how scientific knowledge advances. It illustrates how new discoveries can challenge existing paradigms and open up new fields of study. It also highlights the importance of basic research, studies that might not have immediate practical applications but that deepen our understanding of fundamental biological processes. To conclude, the study of microRNAs has revolutionized our understanding of gene regulation and opened up new frontiers in biology and medicine. As we continue to unravel the mysteries of these tiny but mighty molecules, we're not just advancing scientific knowledge. We're paving the way for new approaches to some of the most pressing challenges in health and biology. Thank you for joining me on this exploration of the fascinating world of microRNAs. I hope this video has sparked your curiosity and given you a deeper appreciation for the intricate and beautiful complexity of life at the molecular level. Whether you're a scientist, a student, or simply someone curious about the world around us, I encourage you to keep exploring, keep questioning, and keep marveling at the wonders that scientific research continues to unveil. If you enjoyed this video, please consider liking, sharing, and subscribing to our channel for more in-depth explorations of cutting-edge scientific research. And don't forget to leave a comment below. We'd love to hear your thoughts on microRNAs or suggestions for future video topics. Until next time, keep curious and keep learning. The world of science is full of wonders waiting to be discovered, and who knows, the next big breakthrough could come from you. Thank you for watching.